Okay, welcome to this ACUR at ANU student presentation session. We acknowledge and celebrate the first Australians on whose traditional lands we meet and pay our respects to the elders past, present and emerging. My name is Rhiannon and I will be chairing today's session. Our first presenter for today is Charmaine Howie, so please start when you're ready.
All right, hello everyone. Hope you're all doing well. Before I start, I'd just like to acknowledge the traditional owners and the custodianship on the lands in which we're all meeting from today. I'm presenting from the UQ St. Lucia campus, so I acknowledge the Jagera and Turrbal people. I pay my respects to their ancestors and their descendants who continue cultural and spiritual connections to country and recognise their valuable contributions to Australian and global society. I'd also like to disclaim that there are images of rock art from the Kimberley region in this presentation. All right, so hello everyone. As I mentioned, my name is Charmaine. I'm an honours student at the University of Queensland and I'm studying archaeology and geography. So my research interests are the intersection between environmental proxies and paleoclimate reconstruction and how this can be used to better understand human interactions and behaviours in the past. So for this particular project, I've been looking at reconstructing the environmental change in the Kimberley for the past 80,000 years. So the Kimberley region is located in the northernmost section of Western Australia, and it is home to tens of thousands of rock art depictions, as well as some of the oldest archaeological sites in Australia. And it's also really beautiful and pretty, and I hope to go there sometime soon as well. So what we know about the Kimberley is that because it's located within the northernmost section of Australia, it's subject to the monsoonal precipitation fluctuations, as well as climate phenomenon associated with the El Nino Southern Oscillation. So these really affect the type of heat transfer and intense monsoonal rainfalls that are on the region. And this affects the type of landscape vegetation and types of plants and animals in the landscape that has evolved to be there. And this would affect the living conditions that people would have had to endure when they arrived there. So we know that people have been in the Kimberley for the past 50,000 years, which is a really long period of time. And we know that people have been in Australia as a whole for the past 65,000 years. So the oldest site in Australia is from a site called Mudgebebi in Arnhem Land, and that's not too far away from the Kimberley as well. But in saying that, despite this region being of cultural and environmental significance, we actually don't have a lot of knowledge about the terrestrial vegetation on the landscape. So usually you can use a combination of terrestrial and marine cores to help understand past changes in the vegetation in the landscape. So in saying that though, the terrestrial cores that we do have only go up to about 14,000 years. And these terrestrial cores are really subject to regional biases. So they give a really good and detailed reconstruction of a really specific area, and they can often have quite different bits of information between the different sites, even if they're quite close together. So they're really subject to this really close site bias. And this is usually where marine cores come in. They provide a more holistic or averaged reflection of the broader environment on that region. However, for the Kimberley, for the marine cores that we have, they're really low in resolution. So a lot of them are collected in the 70s and 80s, and a lot of bits of the actual core themselves are missing and they're really poorly dated. So what we have is this area of environmental and cultural significance and not a lot of knowledge as to how the vegetation and landscape has changed over time. And we have people in this region for 50,000 years. So this is a really big period where we don't have a lot of information for. And why is this an issue? Because it means that we don't know how this vegetation and landscape has responded to more global climate changes, such as changes in sea level, as well as the monsoon and ENSO. So if you look at this illustration in the bottom right-hand corner, we can see during the last glacial maximum about 22,000 years ago, when there would have been people on the landscape, the coastal shoreline was out quite far a bit, so quite different than what it is today. And this would have affected what resources were available, as well as the broader climate conditions that people would have endured. So without this broader understanding, we actually have no context to interpret the rock art and archaeology in this region. And this is important because the ages of the rock art itself are also being pushed back further and further. Recently this year, we had some rock art dates come out at 17,000 years old. And it's more than likely that a lot of the rock art is going to be pushed back even further as well. So our objective is to use this marine sediment core to provide this environmental reconstruction for the past 80,000 years. So we actually want to look at just prior to human arrival as well. And that's going to give us a good understanding of what the vegetation was like on the landscape before people were introduced. But how do we get there? So this is our core site. So the core is located in between Western Australia and this Indonesian island called Sumba. And it was collected in 2016 by this research vessel, the International Ocean Drilling Program. And they do some initial processing on the ship and some scanning. So this is what the core looks like. And then these are samples that we get. So they take out certain chunks down the core for the depths and ages that we're interested in. And then I've been processing them in the lab. And then what we end up is pollen. We end up with a pollen concentrate and some charcoal and organic matter that's been extracted from the sediment on these glass slides. And then comes the fun bit. I get to look at these under the microscope. So this is the methods for how we're going to reconstruct this climate and these paleoclimate records. So we look at the pollen and the microcharcoal primarily. So with the pollen, we know that certain species and plant types have preferential climate conditions that they prefer. Some are more shrubby and prefer different heats and temperatures and wetter or drier conditions. And we know that because a lot of the plants that we see today exhibit that as well. And then we know that these species have different morphology types. So we can go through the glass slides, count how many of each different species there is at different points in time. And this gives us an indication of changing environments. And then we have our microcharcoal. So we essentially do the same thing. We're just counting the concentrations of them. And we're just counting the amount for the charcoal, not the different changes, not the different types, because there's only the microcharcoal. And then this is getting constrained to tell us how old these bits of material are. So we're using oxygen isotope data to tell us the sea level and temperature of these sediments. And we're also hoping to get some radiocarbon dates for our pollen concentrate, which has been very significantly delayed by COVID. But we do have our initial pollen results. So this is the first 10,000 years. And again, this is just some preliminary data. Not a huge amount of information or trends quite yet, but you can see as an example, you've got your eucalyptus and melaleuca, very similar growing habits. Probably if you're from Australia, you probably have seen these a lot before. And you can already see that based on the pollen counts that we have already, you've got increases further down we go, and then closer to where we are now in time, we've got them decreasing. And you've got a similar sort of trend for our mangroves as well. So these species over here are our mangroves. And so this is essentially how our pollen diagrams work. And you can match this up with your microcharcoal concentration and oxygen isotope values. And that's just an example of the similarities between your different species and how this can be used. So the next steps for me with this project are to finish up the pollen samples, finish counting them on the microscope. And because this project is also interdisciplinary sort of focus, so looking at the rock art, we really want to match up the what we have for the archaeological information, so changes that we know have been written about or recognized within the rock art based on different style and different contents, and see if this is aligning with our knowledge of changes in vegetation types as well. So I'd like to acknowledge and thank my supervisors, so Patrick Moss and Helen Bostock from ACUR, as well as our ANSTO partner, Quan Hua. And I'd also like to thank our supporters, Rock Art Australia and the Australian Research Council, and one of the lovely ladies in the lab who has helped me out a lot, Nisha. So that is all from me, and thank you for listening.
Thank you, Charmaine. There are now 5 minutes for Q&A. Judges, do you have any questions?
Perhaps I could start. Charmaine, that was a very interesting presentation. Thank you, particularly as I'm not an expert in the area, but it did grab me. Thank you. It was very clear. I had a couple of questions. The first one is that your analysis is entirely dependent on the integrity of the source material. And so I wondered whether you could just comment on how confident you are that in the extraction process that the source material is preserved, it's not manipulated in any way. And the second question I had for you was, in the process of counting, how much subjectivity is there in that process? To try and objectify that, can it be automated rather than you using subjective measures, as I understand you are?
Yeah, I'm actually really glad that you asked those questions, because those are things that I guess I didn't really consider before I started my honours, and through the process of doing it, these are things that I really want to comment on. So I'll address your second question first. So the subjectivity of the pollen. Yeah, I think this is kind of an issue for pollen counting in general. So there are broader databases that have your pollen types, and you can go and look them up. I get my supervisor to check all of my pollen because I'm not getting huge amounts at this stage yet. So I just take photos of everything and he double checks that. So he's been in the industry for a really long time and he can recognize things super quickly. But I think there is some level of subjectivity, like some things are a little bit on the border, in which case I think there is a little bit of using your best knowledge. Like if it's between two species that look alike, but one only occurs in this area, then, you know, you kind of use that background information. But in terms of the specificity of the morphology, it is like there are pretty huge, yeah, as I mentioned before, there are pretty huge databases. So I think, and it's very broadly used as well. So I think I'm fairly confident in that. In terms of your first question, yeah, there is the material itself. So my material is between like the Indonesian islands and the Kimberley, and we're using it as a proxy for the Kimberley, but we are starting to recognize some things that are from Indonesia and some of the Asian areas as well. So one thing that I want to explore in my honours thesis is sort of how reflective is this of the Kimberley itself, and how much are we getting in from other sites, and also the process itself. When we start off in the lab, we put in a tablet that has an exact number, or it's like 1,000 spores of a pollen that doesn't occur in that area. So throughout the process, if you lose certain amounts of pollen, they use that as an equation because you count how many of that pollen there is as well. So if it's the theory that like if you lose all of like that much, then it kind of evens out. But even the distribution as well, like it's never 100% perfect, and I think like that's just the nature of how these things work. Yeah.
Any other questions from the judges?
I was going to ask a couple of the same questions, but something along the same kind of lines, just in terms of your analysis and when you're quantifying these images using the microscope. You obviously don't do these in triplicate or anything, because it seems like there are a number of samples. So would another person also analyze them at the same time as you to give a bit more assurance as to avoid personal bias? I know that you said that your supervisor's checking them, so would he be the second person?
Yeah, he is the second person, and there's also other people in the lab that I often get over to come have a look. But I like the microscopes are pretty handy. I just take photos of everything unless I'm like 100% sure that that is that thing. And I have a huge reference collection as well, but it's definitely a huge learning process. You have to go back and redo slides that you did at the beginning because you know you get an eye in for it as well. But yeah, just photos of everything. That's the rule of thumb. Record everything, photos of everything.
Thank you, judges, for your questions, and Charmaine for that presentation. We might move on to our next presenter, which is Sabrina Morrison. So please start whenever you're ready.
Okay, thank you. Okay, good afternoon. My name is Sabrina, and I think all of you here today will be familiar with the crop I'm going to be talking about. Most of us wear it every single day, and it is the world's most important natural textile. Of course, I'm talking about cotton. So in Australia, the cotton industry generates over $2 billion annually through exports. And furthermore, it employs over 12,000 people nationwide. So this makes it a very important crop to our national economy. Now, I'd like to introduce you to a fungus called Verticillium dahliae. So Verticillium dahliae causes a devastating disease in cotton known as Verticillium wilt. And basically, verticillium lives out a large proportion of its life cycle existing as these hardy melanized structures known as microsclerotia that basically lie dormant in the soils until they detect the roots of a host plant. This is what verticillium does to the stems of cotton that it infects. So as you can see, it's quite a devastating disease. So why does verticillium cause such a big problem? Well, first of all, it can cause wide-scale epidemics, and because it infects the entire host and usually induces plant death, we often see it causing cotton yield losses of up to 30% under the right conditions. Another issue is that these Verticillium microsclerotia are incredibly resilient. In fact, they can survive in soils for up to 14 years, and this makes it really difficult to eradicate once it's become established in soils. And finally, the fungus has a really broad host range. In fact, this species can infect over 400 plant species worldwide. And this is particularly problematic because it means that even if we remove cotton from the field, the fungus still has the potential to multiply in other plant hosts that it colonizes. Now, the verticillium problem in Australia is quite interesting, but just before I talk about that, I need to explain a concept called vegetative compatibility grouping. Simply put, VCGs are just another way to categorize V. dahliae beyond the species level. And basically, two populations of verticillium are considered members of the same VCG if they can fuse together and exchange genetic information. So in Australia, Verticillium dahliae was first identified in 1957 as a VCG, which would later become known as VCG4B. And for a really long time, we thought that this was the only VCG that was located in Australia. However, in 2014, we identified another VCG called VCG2A, and this led to a wide-scale investigation into our historical cotton collections, which eventually led to the discovery of yet another VCG in 2016 called VCG1A. Now, this is where it gets interesting because internationally, VCG1A is the strain that causes a lot of the devastating yield losses. However, in Australia, it's actually VCG2A that is responsible for a lot of the disease that we're currently seeing. So obviously with these two new strains, we want to know how they interact with Australian ecosystems. And specifically, we want to know things like how are they transmitted from field to field and what kind of susceptible host do they colonize? So this brings me to my research project, which asks two questions. First of all, do either of these new strains infect the seeds of Australian cotton cultivars? And I wanted to answer this question because seeds are potential sources for long distance transmission of the fungus. And if you harvest infected seed and export it to somewhere else, you risk introducing the pathogen to a new environment. Secondly, I wanted to investigate potential alternate hosts for these new strains, specifically looking at weed species that are common to cotton growing regions in Australia. Because as I said before, if the fungus has multiple hosts, it can potentially multiply in the field, even in the absence of cotton. So what I did first is I transformed VCG1A and 2A with fluorescing genes. And the reason we do this is so that when we emit certain lights onto the strains, they glow or fluoresce. And this basically allows us to see the strains colonizing and infecting inside the plant tissues. So we use two fluorescing proteins. One is called the green fluorescing protein, which comes from a jellyfish, and the other is a red fluorescing protein called M-cherry, which comes from a sea anemone. And briefly, I put these two genes into VCG1A and VCG2A, and then use these new fluorescing strains to infect cotton flowers and weed species, and then analyze the infection patterns of these under the microscope. Well, these methods that we used to transform these strains were actually really successful. And we actually created over 50 transformants of these fluorescing strains. So now we had to go along and choose just one of these strains to use in our future experiments. So how do we choose which strain to use? Well, first of all, we want our transformant strains to closely match the morphology of our parent strains. So we looked at things, for example, like growth rate of the transformant strains. And as you can see here in this video, when you compare some of our transformant strains, they were actually very similar to the growth rate of our parent VCG strains. And then secondly, we also wanted to make sure that the disease that was caused was the same as the disease caused by the parent strain. So we had a look at the disease levels, and you can see just by looking at these plants that two of the transformants, 69 and 73, had very similar disease severity to the parent strain. And when you compare the disease score as well, you can see that these were almost identical in terms of disease that was caused. And furthermore, when we look and check these strains under the microscope, you can actually see that transformant 69 is actively colonizing the vasculature of cotton seedlings. So finally, we wanted to look at the fluorescence intensity of these new transformant strains. And obviously, we want to pick a transformant that fluoresces as brightly as possible. And so just by looking at these, you could see that GWT69 fluoresced significantly more than any of the other strains. So when we did our experiments, we decided on GWT69 for our experiments. So now we had both fluorescing strains, and it was now time to infect our plants to see what would actually happen. So to test whether the fungus could infect cottonseed, we used two Australian cotton varieties and infected them with the fungus just below the site of the flowers and waited for seed to set. When we looked at the seeds, we found no evidence of the fungus whatsoever. However, we also took samples from the stems just below the site of seed development, and we did see evidence that the fungus had migrated. So this image on the bottom here shows a technique called gel electrophoresis. And all we need to take away from this image is that each of these bright bands here indicate a stem sample where we successfully found Verticillium dahliae.
Rhiannon, has Sabrina frozen?
Yeah, I believe she has. We'll just give it a couple of minutes and see whether she comes back. Okay, we'll just give it a couple of seconds, see whether she pops back on.
Oh my gosh, I'm so sorry. I have no idea what happened. I just dropped out.
That's okay. Did you want to pick up from where you were?
Yeah, is that okay? I'm
Of
so
course.
sorry.
No, not your fault. It's the Zoom era.
Yeah. Oh man.
We were on the PCR gel.
On the PCR gel. Okay. Cool. Did you guys manage to hear my explanation of the PCR gel?
No.
No. Okay. So what I was saying was that we didn't see any of the Verticillium dahliae within the seeds, but we also tested the stems just below the flowering site or the site of seed development. And basically what we did find was that Verticillium dahliae had migrated further up the stem of the cotton plants. So what this image here is this just shows an image of something called gel electrophoresis. And basically all that we need to take away from this is that each of these bright bands here indicate a stem sample where we successfully found Verticillium dahliae. And then furthermore, when we looked under the microscope, we were able to see that Verticillium dahliae had migrated into the stems of the cotton plants and had begun to colonize the vascular bundles. So these findings suggest that Verticillium dahliae VCGs 1A and 2A lack the capacity to infect Australian cottonseed, but do migrate through the plant. And with this being said, the environmental conditions play a huge role in disease development. And obviously these cotton plants were grown in the most ideal conditions. So a question that remains to be answered is, would this change if the plant were under some kind of environmental stress? So next we looked at our weeds and we managed to recover Verticillium dahliae from 3 weed species. In Nicotiana benthamiana, which is a native species of tobacco, we found both VCG1A and VCG2A cause disease in the plants. However, in liver seedgrass and common sow thistle, we only recovered VCG2A. And what's interesting is that when we look at the disease scores of these species, we see no significant differences between the VCG2A infected plants and the nil plants, which were just treated with water. And this means that VCG2A is colonizing these plants without causing disease. And this is important because it means that VCG2A could potentially multiply in these plants and we would never know. So in conclusion, from my research, we've seen that both new Verticillium strains are capable of infecting mature cotton plants, but they don't seem to successfully enter the seed, which is good because it reduces the chances of transmitting Verticillium into other fields. With that being said, further research does need to be done on whether this changes under different environmental conditions. And secondly, we've shown that these new strains can use weed species as alternate hosts, and importantly, VCG2A can infect hosts asymptomatically, which is something that we need to consider moving forward in terms of disease management strategies in our cotton industry. So thank you for your attention, and I'd like to acknowledge my supervisor, Elizabeth Aiken, and co-supervisor, Don Gardner, as well as Andrew Urquhart for his help and his patience. Thank you.
Great, thanks so much, Sabrina, for that presentation, also for a very well handled technical problem. And I will pass over to our judges, so 5 minutes of Q&A if they would like to jump in now.
Sabrina, oh, go ahead, Ace.
Oh, okay, thank you. I just wanted to say that was a very nice talk. You explained everything really well. I just had a question regarding, sure, you didn't manage to infect the seed. Can you speculate as to why? Do you have any reasons or any hypotheses why this was the case?
Yeah, absolutely. So I think if I to this image here, so the disease triangle, and basically disease severity is dictated by 3 things. We need a susceptible host, we need a pathogen, and we need a favorable environment. And so some of the literature seems to suggest that Verticillium dahliae really thrives when cotton is under stress, or when the cotton plant is under stress, which kind of makes sense when we're more susceptible to disease. So obviously, when I'm conducting my experiments, the cotton is really well watered. It's getting fertilized every couple of weeks. So what I think would be
We'll see whether she comes back. Alternatively, judges, if it's okay with you, it might be best to move on to Nellie's presentation, then do the Q&A for both at the end. Is everyone happy with that?
Yep, that works.
Okay, great. So our next presenter is Nellie Pease. You can go whenever you're ready.
Thanks, Rhiannon. I'll just share my screen. Alrighty, can you all see my screen? And you can all hear me fine? Alrighty, so hi everyone, great to be here. My name is Nellie. I am also an honours student at UQ, but I'm doing my project in paleontology. So today I'm going to talk to you about this very strange extinct animal you see here. Before I start though, I'd like to acknowledge the Yagara people whose country I'm on today, and also the Jaraware people whose country I collected most of my data from for this study. No one alive has ever seen this animal, but there's a very good chance that the ancestors of these people did and lived alongside it, and probably knew a lot more about it than I can ever hope to. So that said, let's talk about Palaeosaurus. This is one of Australia's marsupial megafauna. So you might be familiar with things like Diprotodon, that's this 2-ton wombat relative here, or the marsupial lion, Thylacoleo. These are essentially Australia's equivalents of like mammoths and saber-toothed tigers. Except that we know far, far, far less about them, and in fact about pretty much everything to do with Australian prehistory. And Palaeosaurus, this animal, might be one of the most mysterious of them all. So for a long time we called this the marsupial tapir because we thought that it had kind of tapir-like nose like this. We now know that's probably not true, but it does have a lot of really, really weird features in its body. So I'm just going to highlight a few of those here. We don't think it had a trunk anymore, but we do think it might have had like a prehensile tongue, something like an anteater. Except that its teeth look like leaf eater teeth, like a kangaroo. It has these enormous claws though, like giant claws like the size of your palm, like a bear, and these insanely muscular forearms that would have been almost more muscle than bone. Except that it couldn't bend its elbow more than about 50 degrees. It has this really weird stiff elbow joint. That means it probably would have had to walk with like its arms kind of splayed either side of its body, kind of like a lizard. So essentially this is a bit of a mess. Like this made Palaeosaurus a bit of a paleontological enigma for a really long time. And it's been reconstructed so many different ways as more and more fossils have come to light. I want to draw your attention to one in particular. This is a favorite of mine. This is a little plastic figurine Palaeosaurus from a Cadbury Yowie. I don't know if you guys remember those from like 20 years ago. And it came with this little info booklet that I think sums up my project really, really well. So this is Palaeosaurus. It's big, it's weird, and no one knows for sure what it ate. And therein lies my research question. So I'm trying to see if we can start to pull apart what this thing might have been eating. And obviously people have guessed at this. So this says maybe they were eating roots. Other people have said they might have been standing on their back legs, eating, like reaching for high leaves or stripping bark off trees with these really weird claws, or possibly dredging up like aquatic grasses. But no one's ever been able to test this. And that's really hard because you can imagine why, like Palaeosaurus died out 40,000 years ago with no modern descendants. And it looks so weird that there's really nothing alive we can compare it to. And whatever way it was getting its food, there's probably nothing alive that's doing that now. So this is what we want to know. And it seems like kind of a superficial question, but actually it's really complex in terms of ecology. So animals spend a lot of their time eating. So knowing what something ate tells you a fair bit about how it spent its time behaviorally, and also what its job was in its ecosystem. Palaeosaurus was also a really big animal that was distributed across a lot of Australia. And so whatever it was eating, there must have been a lot of it around. And that can help us start to reconstruct the kind of ecosystems and environments that might have existed in the past. And like I said, we really don't know a lot about these ecosystems. So this is a really important first step in starting to understand this history that we can't see anymore. So how do we do this? A few different ways. So a lot going on here. We've got a lot of methods. The first one is we're looking at tooth wear patterns. And essentially this looks at how the teeth have worn down over the course of an animal's life. That gives you an idea of the texture of its food. So you can really differentiate between tough, fibrous things like grasses or brittle things like bark or maybe leaves and twigs. And then we're also doing stable isotope analysis of a few different elements in the tooth enamel. This is pretty technical stuff. You can find out a lot of really interesting things. So nitrogen is a good way of determining how much meat versus plant was in the diet. Different carbon ratios can sort of align with different types of plants that Palaeosaurus might have been eating. And I don't want to go too much in depth about this, mostly because we don't actually have this data yet. Unfortunately, we're expecting it literally at the end of the week. So this talk is happening about 2 days too early for me to actually show any results. So to be a little bit more interesting, I thought instead I would show you the specimens that we're working with. So to do this project, we went to the Queensland Museum where they have this enormous collection of Palaeosaurus. And I say enormous, it's like 30, which is not a huge sample size in general, but huge for paleontology. A lot of them are just isolated teeth like this. We've also got really nice jaws and skulls down here, which are really cool. We do also have access to a few specimens at UQ, and I can actually show you those in person if they show up on the screen. So this first one here, part of a skull of a sheep-sized Palaeosaurus. This is from about 3.5 million years ago. And if you look, it's kind of hard to see here, you can see how worn down the teeth are. So this animal would have been quite elderly when it died. Compare that to this one on the right here, this little triangle tooth. It's actually a milk tooth. So this Palaeosaurus would have died as like a teenager or the equivalent of a teenager. So we've got a really, really great spread actually of fossils in this collection. And overall, we've got about 30 specimens at a bunch of different stages of life. They're all from this region here, the Darling Downs, and that covers a really long period of prehistory. So the oldest are about 3.5 million, and they go right up until we think maybe 10 to 100,000, sorry, a few tens to hundreds of thousands of years old, which covers the time that humans arrived in Australia, a lot of climatic shifts. So we're not just looking at what one Palaeosaurus ate, we're trying to reconstruct those patterns and of diet, how they changed over a lifetime, how they changed over evolutionary time, and between a few different species, because we think we have definitely at least 2 and possibly 3 different Palaeosaurus species in this collection. We're still trying to figure out exactly which species is which, but that's kind of the next step. So this is really fun research. I get to do cool stuff like squirt this blue dentist putty onto the teeth to make molds to do these wear analyses. And then for isotopes, we drill the teeth, get the little powders out, and we transport them in our very high-tech freezer setup to the mass spectrometer that measures our ratios. So it's really fun. So to speak to the theme of the conference, that's my search, but what does this mean for our future? Because really we're kind of talking about the past here, right? If you ask any paleontologist, they will tell you that we need to understand the past before we understand the future. So we really, like I said, don't know a lot about how past ecosystems in Australia work. And to be totally honest with you, we aren't super sure how they work in the present either. Like Australia's ecosystems are really unique in the sense that we don't have any of these large herbivores like Palaeosaurus left compared to places like Africa and the Americas where you have buffalo and bison. We don't have any large carnivores either. And that means that the dynamics that operate on ecosystems in Australia are quite different to how they operate in other continents. And we're not going to understand those and how they might change in the future till we understand their context and the kind of trajectories they're on. And Palaeosaurus covers this period of time leading up to ours really, really well. So we've got samples from the Pleistocene that cover when humans arrived. We can start to answer questions about why they might have gone extinct, what made them vulnerable to that. And the period that's almost more interesting to me actually is the Pliocene. So this is that older period. This is the same age as our really old samples. And the late Pliocene here is actually, we think, the last time in Earth's prehistory that temperatures and atmospheric carbon concentrations approach what we think they will be at 2100. So it's kind of this perfect little geological testing ground to see how ecosystems respond to conditions like this. The issue is, though, there's not a lot of sites in Australia that preserve that period. And actually the best one we have is Chinchilla, which is the site that this specimen here is from. So with this study, we're hoping to see if we can kind of reconstruct the ecology of this site and this period in more resolution than we've ever seen it before. Look at how this one animal responded to changes that were happening, how it evolved in response to these changes, and what kind of changes were eventually too much for it to deal with. This is sort of a really preliminary first step. We don't expect to solve these, I guess, answer these questions completely with this one study. But like I said, we don't know a lot. And so this first step is a really important one to take. Just to speak to Palaeosaurus in particular, one interesting thing that I want to point out is a lot of the theories we have for how Palaeosaurus got its food involve really considerable trauma to plant communities. So we're talking about, digging up roots or pushing over tree ferns or stripping bark off trees. And that's actually quite similar to what we see modern elephants doing now. And we talk about elephants as niche constructors. So they're really important for clearing forests. They actually kind of open up savannas and change the structure of forests by foraging in them. And it's not a huge leap to suggest that Palaeosaurus might have been doing something similar in Australia. And that is really interesting if you think about what our ecosystems are like now without this animal. So for 8 million years, we had Palaeosaurus here engineering the sort of structure of our forests, and then 40,000 years ago, it disappeared. And what did that do to ecosystems since then? So again, our study's not going to be enough to answer that question entirely, and it's really pure speculation until we can get some data and really a lot more studies on this period. But this is the best, most quantitative study on Palaeosaurus so far, and we hope that it's a really interesting first step. So I'd like to acknowledge these two societies for giving me funding for this project, and also my supervisors and collaborators. Hopefully soon I'll have some more exciting data to share. Thank you very much.
Great, thank you, Nellie. We might go straight into your Q&A just while it's still fresh, and then we'll take 5 minutes to do Sabrina's as well. I think it might be best if Sabrina keeps her video off just to try to prevent any further connection issues. Yeah, judges, any questions for Nellie, please go ahead.
Nellie, I really enjoyed that presentation, I have to say. It was like a show and tell.
I feel like that every time I talk about it. I'm like, here's my favorite dinosaur.
Yeah. So look, I wondered whether you had any preliminary data from the analysis that you're expecting data from in the next few days. I mean, presumably you would have use that methodology to validate some of the early samples. Of particular interest is the role of nitrogen in differentiating between meat and vegetation consumption, and whether or not you think that there's a role for predation in this.
Yeah, really good question. We don't expect that there will be any role for predation at all. What we'd be looking for there is eating insects. I think I mentioned in the body reconstruction, actually you can kind of see here, we think based on the shape of the jaw that it had a really long prehensile tongue. And it's also got these really big claws and really strong arms, which are kind of things that you tend to see in ant-eating animals or termite-eating animals. We think that's really doubtful, just because it's got these really, really chunky cheek teeth, these molars, and you don't need those if you're eating insects. Most insect eaters don't have teeth at all. So essentially this is, we're expecting to get a negative result here. We just want to test it because a few people have kind of thrown around this idea like maybe it was eating termites, but no one's actually proven it. And the way you test that is, so like I said, nitrogen, the nitrogen, different isotopes of nitrogen 14 and 15 fractionate differently in plants versus animals. So you'll get a really enriched nitrogen signature for something that's eating a lot of protein, and a depleted one for something that's eating only plants, and kind of intermediate if it's a mix. No one has, you mentioned preliminary results, no one's done this on Palaeosaurus yet. My supervisor has done a few studies on some other megafauna, so we know it works, and we get sort of really consistent, results that match with the things that look like carnivores have a meat-eating signature, the things that look like herbivores have a plant-eating signature, and it's actually a pretty established analysis in ecology in general. So, yeah, the issue with that is the age of our specimens, because nitrogen depends on there being protein left, and we don't think there'll be any in the older specimens, but maybe the younger ones. It'll be interesting to see. Preliminary results wise though, we don't have any data for our specimens, but some of our collaborators have tested Palaeosaurus before, and I was kind of anticipating a question about isotopes, and so I've got this graph. This is from a previous study at the site, and we expect that isotope wise, based on one specimen that was analysed like 10 years ago, it's probably going to plot out somewhere here in terms of the carbon isotopes, and that's a leaf eating signature. We think it's probably a leaf eater, just based on morphology and a lot of things, but it'll be interesting to see. Sorry, that was kind of a long answer, but yeah.
Thank you, Nellie.
No worries.
Okay, I might have time for one question for Sabrina, and then I do need to give this link over to the next presentation. But if anyone could go ahead, that would be great.
Sorry, Rhiannon, you wanted a question for Sabrina?
Yeah, sorry, because you didn't get a chance for her Q&A before. So
Yeah.
if she could have an opportunity to answer a question now, and then we will end this session.
Yeah, Sabrina, look, I wondered if you could tell us something about the genetic similarity between 2A and 1A. I wasn't quite sure how similar the two species were. And second, how were you actually measuring disease? It seemed to me from your presentation that it was visual inspection. Is there something a bit more sort of objective or quantitative than that? So really, differences and similarities between 1A and 2A at a genetic level, and second, how were you measuring disease?
I think we may have lost her. But thank you very much for your question regardless. I think that might have to bring us to the end of today's session. So thank you to our judges, Dr. Zengin and Professor Kashigian, and congratulations to our student presenters. You all did a really great job. This recording will be made available online following the conference, and we hope to see you in other conference sessions this week. So thank you, everyone.
Thank you.